Lung cancer is the #1 cause of cancer-related deaths in the US and worldwide. It's also the most common type of invasive cancer, estimated to cause 154,000 deaths in the US this year. With a 5-year relative survival rate of 18%, things don't look great. There are two main types of lung cancer: small cell lung carcinomas and non-small cell lung carcinomas. Small cell lung carcinomas are generally treated with chemotherapy and radiation, and although the cancer often comes back eventually, a large number of patients go into remission. When we look at non-small cell lung carcinomas, which make up 85% of all lung cancers, we see that relatively they are much less responsive to chemo and radiation, and therefore rely on surgical resection, pre- and post-op chemo, as well as targeted and immunotherapies for treatment. Even with these treatments, the 18% survival rate still stands. However, thanks to the completion of the Human Genome Project, as well as recent advancements within fields such as proteomics, molecular diagnostics, and pharmacogenomics, we're getting closer and closer to defining the historically unpromising future of treating lung cancer. And so, this is the story of the future. In 2007, a group of Japanese scientists and a group of our very own CST scientists simultaneously but independently discovered the EML4-ALK fusion gene, which was found to cause cancer growth in non-small cell lung cancer. How does this work? Well, basically, it plays into the fact that all cancers are a result of gene mutations that cause abnormalities in cell functions. In this case, the gene rearrangement fuses the protein EML4 together with ALK, a receptor tyrosine kinase, which usually isn't expressed in the lung. The ALK fusion gene, even when fused with other genes besides EML4, activates the coding of tyrosine kinase, which inhibits cell apoptosis, meaning that the normal, controlled death of cells is no longer regulated, leading to tumor growth and malignancy. The ALK fusion genes are responsible for around 3 to 5% of non-small cell lung cancers, more prevalent in adenocarcinomas. If we could somehow stop this uncontrolled cancer growth, we would be giving our lung cancer patients a lot more time to live, and our scientists a lot more time to find a cure. This is where Pfizer comes in. Using the findings published by our CST scientists and SODA's group, Pfizer developed an ALK inhibitor drug called crizotinib, approved in 2011 by the FDA. This was a miracle drug. It stopped the kinase activity that caused cancer cell growth. This first-generation drug changed the game drastically. Before, people who had tried and failed chemotherapy had a response rate of around 10% and an average progression-free survival of 3 months. Crizotinib extended that to a 50 to 60% response rate and 7 to 10 months of progression-free survival. Promising, right? Even more so are the second, third, and fourth generation medications that have been developed and approved by the FDA or are currently in the process. These are important to the drug resistance that almost inevitably develops over time, so patients who were initially treated with crizotinib still have a chance. Tyrosine kinase inhibitors give us time, an inarguably critical factor in the battle against any cancer. By keeping the tumors in check, they open up a plethora of new possibilities in treatment, and with the development of these targeted therapies, we are not only one step closer to giving lung cancer patients a more promising chance, we are also one step closer to beating the battle against cancer.